So I'm still actually soaked in the water of the first wave of Stefano, and I will add a few drops to a kind of strongly connected topic. So first of all, let me thank all the people involved in this kind of large collaboration. Many of them provided different contributions to this work. And I mean, many of them you probably know. Okay, so given the position of my talk in this session, I think I will skip most of this introduction, talking about motivation, about yielding, why mayonnaise can be considered as a solid, but also a liquid, and what happens in between. I think it's a little bit not necessary. What I would like to stress is a point that is already made by many speakers, is the fact that experimentally and also theoretically, investigating yielding is particularly difficult, in particular in amorphous material, given the multi-scale nature of the problem. So there are at least 3 different levels of description. One is the macroscopic one, where yielding can be identified as a change in the rheological mechanical response of a material. There is something like a mesoscopic scale, which is also relevant. So the fact that once stress is imposed on a material, the deformation can be not uniform, but you can have spatial structures within the material. And then there is the elementary microscopic level of description, so which are the elementary process mediating the onset of plasticity in complex material. And there are many outstanding questions. Many of them were already pointed out by Stefano about the universality of this concept. If we consider yielding a sort of phase transition of some kind, if there is some critical behavior that can be associated to yielding and so on, just to be clear, I will not answer address any of these questions. What I'm going to do is to provide my contribution in terms of the experimental results that we obtain on a few selected systems with an experimental approach which is somehow similar and somehow complementary compared to the one just presented by Stefano. So our approach is based on the use of a stress-controlled shear cell, which is based on a design proposed by Stefano a few years ago. It is being optimized to be coupled with a commercial inverted microscope. So we have 2 parallel plates, transparent, where the sample can be squeezed in. The gap can be tuned between some tens of micron up to a few millimeters. And the sample can be observed from below using a commercial bright field microscope. And we can apply arbitrary stress profile to the sample. And typically, we can go down to shear stresses of about 1 Pascal. So relatively small forces can be applied. And what I would like to point out is that even with this relatively simple setup, we are sort of able to bridging between these 3 length scales. So we can have a look on the macroscopic rheological response of the sample. We can have a kind of precise idea of how the strain is distributed across the gap. And can we also monitor what happens at the elementary scale in terms of particle displacement and rearrangement. So we are going to present results obtained with mainly 2 samples. One is very, again, quite similar to the one used by Stefano. It's a microgel dense suspension. And then we also present results obtained with a dense oil in water emulsion. In both cases, these are samples obtained by squeezing particle with a size of the order of 1 micron. They are both optically transparent, and so we can have a good optical access to the sample, and are highly mechanical reproducible. So we can restore the material properties after each experiment. So what I'm going to present are actually only oscillatory shear experiment, where we apply a sinusoidal stress profile on our sample, and we monitor in a steady state condition what's going on. And typically what we do is we keep constant the frequency and we perform an amplitude sweep. So the first thing that we can do is to apply our oscillating force and look at the sample. What we see is this. Now the sample is pretty transparent, so we are adding some tracer particle to better visualize the displacement. So what you see is basically the sample going back and forth as we shear, but we can measure quite accurately this displacement. And so while we measure when we know which stress we are applying, we can measure the actual deformation. And so we have simultaneous access to stress and strain, and so we can do rheology, basically. So we can measure G prime, G double prime, or also obtain Lissajous figure. And I mean, at least in a certain range of amplitude and frequency, this works more or less like a rheometer. Then we can also exploit the finite depth of focus of the microscope to image different plane across the gap, so we can have space-resolved information on the amplitude of the deformation in different plane, with a decent actual resolution of about 20 micron or so. So for example, in this case, what I'm plotting here is the amplitude of the oscillation in different position across the gap, for a different value of the imposed stress amplitude. And from this kind of amplitude profile, we can evaluate the local strain, basically the relative displacement of two neighboring layers within our sample. As you can see, in this case, it's pretty homogeneous. There is a little bit of slip close to the bottom, but for the rest, the overall strain profile is pretty ideal. So it's rather homogeneous. So we can measure the mechanical response, we can probe the deformation profile, and the last thing we can do is to have an idea of what's going on at the particle level. To do so, we use the ECO protocol. So basically, we take picture of the sample at a distance which is equal to the period of the oscillation. And the idea is that if all is, or what is going on the sample is completely reversible, then after each period we're back to exactly the same position. If something irreversible has occurred, we should be able to relatively easily detect. So this is an example of an echo movie recorded for a shear amplitude well below microscopic yielding. And so all pictures looks more or less the same. There is nothing interesting going on. But if we increase the amplitude of the deformation, what we see is that the particles start showing some sort of effective dynamics. So a sort of shear-induced mobility, okay? And we can visualize this kind of plastic events using an approach similar to the one presented by Mathieu, taking differences between pair of consecutive images and obtain this sort of activity maps. Actually, since we have particles that are well above the resolution of the microscope, we can do more. Actually, we can accurately characterize these effective dynamics. We did this with different methods. For the sake of this presentation, I will focus on results obtained by single particle tracking. So by finding the particle, this tracer particle in each frame, and follow the trajectory and characterize the dynamics. So it turns out that the shear-induced dynamics can be thought as the superposition, as also Stefano was presenting, of two well-separated contributions. So even a small applied strain, there is something that looks like a flow. I mean, it's not an actual flow, but it's something like a highly spatially correlated, very slow, highly persistent motion of the whole, across the whole field of view. Okay? Something like a ballistic contribution to the dynamics, which is constant for the whole duration of the experiment, highly temporally persistent and spatially correlated. On top of which, there is some random positional fluctuation exhibited by the particle. So we can, for each imposed stress, we can accurately characterize the spatial distribution of this sort of slow flow field, which is more or less always present, but I will not focus on this contribution. I'm more interested in, is there a question?
Come back. So maybe I'll ask it now. Yeah. So here you shear, you stop, and then look at the particle.
I keep on shearing, and I take a picture every period. Yes. So macroscopically, every time I'm back in the same position, but if some change has occurred, this is what I'm looking at.
Percentage what they are?
This is the strain. So well below the macroscopic yielding, roughly across the yielding, and well above the macroscopic yielding. So this is the ballistic contribution, which is always on top of the plastic dynamics.
A large contrast, but it's the angle. And what if you show us? Can you get a strain? Can you get a scale of a strain would compare to gamma? So.
Yeah, I mean, this color map is just outlining the degree of alignment of this flow field. So here, the average drift contribution has been already erased, and the purpose of this representation is to show that this motion is long-range correlated. So the correlation length of this flow field largely exceeds the field of view. So what we are seeing is a small part of a large drift, which is all across the sample. Sorry?
You observe a small part of the sample? Or?
A small? A relatively small part of the sample. So yes, the field of view is something like 1 millimeter. The whole sample has a size of about 1 centimeter. We are far from the boundary. I mean, relatively far from the physical boundaries of the sample. Okay, so we can identify this ballistic contribution to the dynamics as a persistent flow-like field, and which can be removed by single particle trajectory relatively easy, given the fact that it's highly persistent. And so let us focus on the single particle stochastic dynamics, and what I'm showing here is the mean square displacement of the particle for different applied stress. And in all cases, the mean square displacement is well described by this simple expression, a constant plus a linear function of delta t. Here, delta t is the number of elapsed frames. Okay? So basically, there is some sort of short time plateau that is associated with this peculiar motion that is displayed by some of the particles, even a relatively small strain, that displays some sort of bistability or multi-stability. So some particles perform jumps that after a certain number of cycles can be reverted back to the original position. And this sort of rattling in the cage motion is what is giving rise to this short time plateau in the mean square displacement. While the long time dynamics is dominated by the rather clean linear scaling. So the fact that the mean square displacement displays a linear scaling enables to characterize it by the single number, which is basically a diffusion coefficient. Actually, since it's a mean square displacement per cycle, the diffusion coefficient will be in micron square per cycle. And here I am representing the value of this diffusion coefficient as a function of the local strain in this case, and you see that there is a huge explosion across the yielding transition. So the effective mobility of the particle increases by orders of magnitude across the transition. So what we are looking at is not the motion of the particle of our system, is the motion of tracers. And you can ask whether this is indicative of the actual dynamics. And so we repeated the experiment using tracers of different sizes, and the mobility that we get is not the same. So this diffusivity depends on the particle size, while the velocity, the mean square velocity associated with this correlated flow doesn't. So it looks like the particles are just dragged by this overall correlated motion, while the random dynamics depends on the particle size. And the dependence on the particle size is just the one you would expect in the case of a Stokes-Einstein-like relation. So if you scale the diffusion coefficient like you would do in a microrheology experiment, so by multiplying by the radius, then you observe a nice scaling, a nice collapse of all the curves. So probably this product D times A reflects some intrinsic properties, or some intrinsic property of the sample. It is not longer dependent on the particular probe particle we are using to visualize and probe these shear-induced dynamics. So far, I presented results obtained for this carbopole sample that represent a sort of, yes.
The size compared to, so yeah, we saw microns.
Yes.
The carbopole itself is,
I mean, in this case, it's about 500 nanometers, roughly. So we are, and this is true also for the case of the other sample that we'll present, we are a little bit below and somehow above the particle size in both cases. So we are never, the tracers are never much smaller than the characteristic size of the sample. So we start from something comparable and then a factor of 3 larger than that. So at least in this regime, the one that we explored, this sort of scaling seems to work nicely.
And the probe is the size of the particle.
Roughly, yes. Okay. So let me spend some time on the other sample, which has something interesting. And the first big difference that we found is that the deformation profile is no longer homogeneous. So the amplitude of the deformation is not a linear function of the height. And this means that the local strain is different in different position across the gap. So there is some sort of strain localization or shear band formation, if you want. So again, what we are looking at is we are under oscillatory shear, and we are looking at, in steady state condition, at the relative displacement of different layers in the sample. This is what gamma local is. Okay, so the microscopic dynamics is very similar to the previous case. Again, the mean square displacement shows a short time plateau, and then the long time dynamics is characterized by a certain effective diffusion coefficient. The big difference is that in this case, the statistics of particle displacement is strongly non-Gaussian. I didn't tell you, but in the previous case, the statistics of particle displacement was rather ideal. So was very close to a Gaussian. So the analogy with Brownian motion was almost perfect in the previous case. Here we have still a clean Brownian-like scaling, but the statistics of displacement is highly non-Gaussian. We'll go back to this property in a couple of slides. what I would like to stress is that given the fact that the local strain is different in different position, we can, if we explore the dynamics in this stratified medium, we find that the dynamics is position dependent. So in a condition where there is a huge difference in local strain between the bottom and the top, we find that the bottom is basically stuck while the microscopic activity increases a lot as we go to the region of larger local strain. And if we plot the mobility of the particle as a function of the local strain as obtained by scanning along the vertical direction the sample, and we superimpose the result obtained by remaining in a fixed position and by increasing the applied stress, then we get a nice collapse. This tells us that this mobility, the shear-induced diffusion, is a function only of the local strain. So, if we compare the rheological response of the two samples that we consider, there are not huge qualitative differences. One of the probably most remarkable difference one can spot is how steep is the increase of G second prime close to the overshoot. So this, I mean, it is the definition I'm reporting here is not exactly the one suggested in the paper of Donnelly, where this concept of viscoplastic fragility was introduced, but still the logarithmic derivative of G second prime, the maximum value of the logarithmic, has been suggested as an indicator of the level of abruptness of the yielding transition. We see that there is a huge difference between these two samples, and this is somehow reflected by how steep is the explosion of the macroscopic dynamic across yielding. Okay, so far is qualitative observation, but it is true that if this yielding transition is smoother, then the increase of the microscopic dynamic is somehow milder compared to the case where the viscoplastic fragility is very large, and in this case we observe even more explosive increase of the microscopic mobility. We also looked at a higher order indicator of how this microscopic dynamic is, and we found that, for example, if we consider the deviation from Gaussian statistics of the displacement, the emulsion display a highly non-Gaussian behavior, which is particularly pronounced across yielding, while this is not the case for the carbopole. And we also looked for the presence of dynamical heterogeneities. In particular, we consider as an indicator for the dynamical susceptibility, for the presence of dynamical heterogeneities, the dynamic susceptibility K4, that is calculated basically as the variance of a suitably defined overlap parameter, and it is an indicator of the degree of cooperativity of the plastic rearrangement. Okay. Again, we are subtracting any ballistic contribution. We are only focusing on the plastic activity. And so we are looking at correlation in the plastic activity, which are not present in the homogeneously yielding sample, while they explode significantly in the shear bending sample. And not only that, we also found a kind of an intriguing correlation at this level between shear bending and dynamical heterogeneity. So what I'm plotting here is, again, for different imposed stresses, the deviation from a uniform strain and as a function of the local strain. So basically, I consider the deformation profile. The derivative of the deformation profile is our local strain, and the second derivative is the deviation from an uniform strain condition. Okay, and actually the deviation from uniformity, if you wish, as a function of the local strain is represented by basically a single curve for all imposed stress. And if we plot the peak of the chi4, so the maximum, the largest value obtained by this indicator of degree of cooperativity of the dynamics, we found a nice colocalization. So I wouldn't talk about causality, but at least there is an interplay, apparently, between the localization of shear bands within the sample and the divergence of this dynamic length scale associated to the cooperativity of plastic rearrangement. Yes?
It's at the interface between the two shear bands. Is that what you're saying?
Yes. So where the deviation from an homogeneous profile is larger, the peak of the chi4, that is the dynamical heterogeneity, is larger.
People are not moving on one side of the interface and some are moving on the other? Or is it more complicated than that?
I would say it's more complicated. I mean, I would say it's less trivial. Hopefully, it's less trivial than that. In particular, because I think that the scale of variation of the strain is larger than our depth of focus. So this is something that we have to check. But what I would say is that we can assume that locally gamma is constant. And so this heterogeneous behavior is not a consequence of the fact that we are mixing between different planes, but this is something intrinsic. But these are absolutely good points, something that has to check carefully.
Yes, so the chi4 is what you're using as a proxy for the dynamic correlation.
Yes.
And,
Sorry, I mixed a couple of things. I talked about the chi4, and then I talked about the correlation length. We also, I'm not sure I have it, but here in the presentation I can show you, we also calculated the G4, so the partial correlation function, and we extracted an estimate for the correlation length of dynamical heterogeneity from there. So what I'm plotting here is just the peak of the chi4.
the previous question, you have extreme heterogeneity because of shear banding. Is that?
I mean, shear band correspond to plane with different local shear, and what we are looking at is the dynamics within each one of these planes. So the modulation across the sample due to shear bending is orthogonal to the plane we are looking at. So we have, Thomas is absolutely right, we have to check that this does not introduce any artifact due to out-of-focus contribution, but in principle, these two effects should be decoupled. This should be a genuine correlation. Okay, I think that with this, some of these results are published, some of them we are still struggling to put them together. So I'll stop here. I'll thank again all the co-authors and you for your attention. Thank you.